Your body's skin cells are a prime example of a cell in the body that goes through mitosis. Mitosis being the process that your cells go through to divide. So you might remember from the lectures that a cell does not grow larger and larger, but it divides. And that's basically for your skin, that's how you're able to get full coverage of your body. Your skin is not one large massive cell, but millions and millions of cells that are continually dividing. One of the prime examples of how your skin cells divide and why they need to divide is because your skin is continually being exposed to external environments, so you constantly need a new layer of skin to keep up with the protection of external stimulus. So, for example, your skin, your face, you're constantly fluffing off dead skin cells to expose brand new cells. Well, those brand new cells underneath are constantly dividing, and they're going through this phase called interphase. Okay, so every cell is going through interphase where the DNA, which is all in here, it looks really messy. So the DNA during interphase is dividing. It's going through its normal processes of growing and cellular respiration and what have you. So as these cells divide, mitosis is the stage in which the cell is preparing itself to divide into another pair of cells. Okay, so when it divides, both cells are genetically identical. Okay, you have the exact same amount of material in the new cell as you did with the old cell, so it makes a complete replica of itself. So as this cell prepares itself for mitosis, or for division, interphase makes an identical copy of the DNA in the nucleus, okay, and it kind of slowly phases its way into prophase. Okay, now just to kind of make this a little easier, one of the acronyms that you should remember, which will make this easier, is PMAT. Okay, this is one of the stages. This is the last stage of interphase. Okay, interphase has three stages. Okay, now we'll talk about those 3 stages in another video, but one of the last stages is mitosis. Okay, and mitosis is broken up into these 4 substages. Okay, first one is going to be prophase, and then metaphase, anaphase. Let me use a different color. Prophase, metaphase, anaphase, and then telophase. Okay, or cytokinesis. Now, in my diagram here, you're going to see that there's an extra step here, ProMetaphase. You don't have to necessarily remember that. Just remember, ProMetaphase is the stage in between prophase here and metaphase. Okay, so you'll understand the need for, or why this particular textbook added this this extra step. So again, we go into prophase. You might notice that the DNA is now a lot more nicely, tightly organized, and it's copied now. So it's been copied. So now you have 2 copies of the DNA, and it's tightly packed and organized now into something called a centromere, okay, or chromosomes. Your chromosomes are what carry your genetic material that are passed on from a parent to a child, or the genetic material that's passed on from one cell to the next cell that's shared. You'll also notice that the spindle apparatus is beginning to take a little bit more form. If you compare it to interphase, where you have the centrioles, well, the centrioles now are beginning to form their microtubules, okay, or their asters. Okay, these microtubules play a very important part in helping to organize the DNA that is in the nucleus. Okay, it helps to organize the DNA, and it's going to later on help to separate the DNA, the copy DNA, from one cell to another. Okay, so just again, a step that you don't necessarily need to remember for test purposes, but it's good to know, is that you'll notice here the difference between our prophase here and then our metaphase is that the nuclear envelope has now disintegrated. Okay, so you have a disintegrated nuclear envelope, and then you have a more clearly defined and organized chromosome. Okay, so you'll notice that these chromosomes also have something called a kinetochore. Okay, these kinetochores are located in the centromere, in the center of the chromosome. So here you have your chromosome. Okay, and then here's the identical copy of it. And then in the middle, you have, in the centromere, you have your kinetochore. Okay, these are proteins that these microtubules will attach to, and are later going to help in evenly dividing up the DNA that is in these chromosomes. Okay, so let me go ahead and erase what we have here, just so we can have a nicer board to look at. So again, just quick review of ProMetaphase. Your nuclear envelope is disintegrating, your chromosomes are a lot more tightly knit and packed, and your spindle apparatus is now connecting itself to the chromosomes and preparing it to be divided. Okay, so you can guess of what's going to be next, what state is going to be next. We had Prophase here, Metaphase. Well, actually, Metaphase will be what's next. After Prophase, we go to Metaphase, which is on in the next video. And before we had Prophase and Metaphase, we had Interphase, which was the preparation for mitosis. Okay, so the next video we're going to finish this up by talking about Metaphase, Anaphase, and Telophase.